Okay, thank you very much. Good afternoon, everybody. Today, Raghu and I will share with you our experience of the creating BIMA package that we had at our latest NDA submission last year. I will give you a brief overlook of the BIMA components and more detailed explanation of the clean site dataset, define PDF and BIMA-RG, while Raghu will spend more time on the approach to generate subject level data listings and on details of the submission of the BIMA components. A bit overview and some background. CEDAR Office of Scientific Investigation has specific responsibility for verifying the integrity of data submitted to CEDAR in support of the application. In order to do so, Office of Regulatory Affairs conduct on-site inspection of clinical investigators, sponsors, CROs, and IRBs. Inspection planning includes selection of clinical sites and other regulated entities that were contracted by sponsors to conduct the preparation of documentation that are provided to inspectors that actually perform the inspection. In the past, applicants have frequently provided this information in different data formats that were not conducive to timely inspection planning and conduct of the inspections. In February of 2018, FDA published a draft guidance on the standardized form of the biosearch monitoring, BIMA. BIMA has three main components as per guidance: clinical site level information, subject level data listings by clinical site, and summary level clinical site dataset. These three components need to be in ECD-CTD format and included in Module 5 of the applications. BIMA study tagging file should be constructed and should be placed in Module 5 in the other study report and related information section. While we started working on the implementation of the guidance, we identified components that we already have available in some form and components that are completely new. So we have existing CSR listings that can be converted into subject level data line listings. We have clinical study level components like clinical site information, protocol, sub, CRFs. But there are a couple components that are brand new and we have to develop them from scratch. This is summary level clinical site data set and defined XML and BIMA reviewers guide. We have to collaborate with other functions in the company, clinical operations and regulatory operations, to get some information that we needed for the data set and for BIMA package. Let's look at each component in details. Clinical study label information consists of listing of sites participated in clinical studies. We have them in appendix in module 5 of CSR. Site identification, this information includes site number, address, and other contact information, name of the clinical investigator. Then we have already in CSR in section 6 a list of sponsors who we contracted to conduct the study. And protocol with all amendments already in section 16.1.1 of module 5. And annotated case report forms are part of ECTD data package. As per technical conformance guide, if these components already exist in CSR, we don't need to resubmit them, but we need to provide links, hyperlinks to those components, and we did it through BIMA reviewers guide that I will talk about later. Another component, main component of the BIMA package is site-level data set. It contains data from all pivotal studies submitted. It is a standalone data set and is independent of the CDISC data models used in the creation of SDTM tabulation and ADAM analysis. It has one record per pivotal study per clinical site per treatment arm. We can identify three categories of the variables in this data set based on the source of the information of those variables. The first one is study level variables. Those variables information comes from protocols, REAGOPS, and statistical analysis plan. Another group of variables are safety, primary efficacy summaries, and disposition variables. This information comes from clinical database. The only exception is screen, which is a number of screened subjects per site. Our database doesn't collect screen failure information, so the information from the database won't be complete. So we use IVRS data to get information about all screened subjects. And the last group of variables is site and investigator details, including financial disclosure amount. This information we got from cleanups function. While working on clean site data set, we come across some cases that we need to figure out how to deal with. We have subjects who transferred between sites during the course of the study. We decided to summarize those subjects under the latest site the subject participated at. We also had satellite sites that were summarized under main site. Sites with only screen failures were presented as single record per site with arm safety and efficacy and disposition variables set to null, and number of subject in safety population and full analysis set population were set to 0. There is only one variable treatment arm as per conformance guide, but we have cases where planned treatment for some subjects was different from actual treatment. So we summarized efficacy and disposition variables excuse me, using planned treatment arm and safety variables using actual treatment arm. We have to create one additional variable which was not part of the technical conformance guide, full analysis set population, to represent planned arm, and safety analysis population was in technical conformance guide, and this is safety population. Some variables in the technical conformance guide had labels which exceeded 40 characters, so we had to adjust those labels to be within 40 characters. Clean site dataset should go with defined PDF. We created defined PDF by converting defined XML into PDF format and created links to clean site XPT dataset and BIMRG. Bioresearch monitoring reviewer's guide is not required but highly recommended. It is a good starting point to understand all BIMA components and study details. There is no industry-developed template, so we developed our own template of this document. I would consider BIMRG as a centerpiece, which connects all BIMA package components together. It has 4 main sections. The first one is introduction that gives description of all BIMA elements and provides hyperlinks to module 5 of the CSR and other components. It also contains overview of study details for all pivotal studies included in submission, details like protocol, study design, treatment arms, dosages, primary case and key secondary endpoints. Another section is clinical study level documents, which provide hyperlinks to site information investigator financial disclosure, all the vendors who were contracted by the sponsor to conduct the study, and study documents like protocol amendments, annotated case report form, and statistical analysis plan. Third part is subject level data line listings by site. This part of BIMA RG contains information about listings covering 10 topics identified by Office of Scientific Investigation, and Raghu will spend more time on details of this. And the last component is a description of a clinical site data set and defined PDF, which includes variable description, structure of the clean site data set, additional details about derivational algorithms that were not described and defined PDF. It has hyperlink to clean site data set location, and give some clarification regarding specific cases that I was talking earlier, and any other information that could benefit the site selection and conduct of inspection. Yeah, this is, I give the screen to Raghu, who will continue and explain more about our approach to subject level data listings and details of the submission of the package.
Thanks, Olga. Can you hear me?
Yes.
Thanks. So I'll briefly touch base about the components that go into the ECTD component for the BIMO package, and then also talk about the details about the creation of the site listings. And for the ECTD component for the BIMO, what does the BIMO draft guidance and technical guidance document says? It says we need to submit this whole BIMO package in the module section 5354 other study reports, and the study ID would be a BIMO. And it asks to include the clean site data set along with the defined.pdf. So that's a guidance suggests to have these components in this section of the ECTD component. So using that as a starting point, we have built our own ECTD package. We followed similarly what the language says in the BIMO draft guidance and the technical conformance guidance. We built a BIMO study tagging file with the BIMO study ID. Then the tagging files for the clean site data set would be the data listing data set along with the data listing data definition for the defined. As Olga just talked about the BIMO reviewers guide, we did include in the study report body components the BIMO reviewers guide, and that provides all the hyperlinks to the different sections of the submission package. Talking about the hyperlinks, we chose to submit our site level data listings within the protocol, within the individual pivotal studies itself, along with the clinical study level data set information. So that we have those individual site level listings within each study, but still we have provided the hyperlinks in the BIMO reviewers guide. So this concludes the BIMO submission package component. And in the next set of slides, I'll talk about the creation of the listings using our CSR listings. So the draft guidance and the technical guidance has a language says like we would need to create this. We need to submit our site subject level data line listings for each major pivotal study that is submitted in the submission component. Even though if they are the same investigator is involved in the multiple studies, we need to provide the site information for that each individual study at the study level. The technical conformance guidance, the technical guidance also says about the options to submit these site level listings, either by going with option A and option B, where the option A says we need to submit within each study by site and the 10 oversight topics, which I'll be talking a little bit later. Or the option B was within each study, within each site, within each subject, you could summarize you could list your all the 10 oversight topics. Since we had an option to go with either one, we chose to go with option A. So what are the 10 oversight topics that were mentioned in the BIMO draft guidance and the conformance guidance? As you can see, these are the pretty much most of those topics are within what we have in our CSR listings: the consented, discontinuation, study population, inclusion, exclusion criteria, adverse events, IPDs, endpoints, efficacy endpoints, conmeds, and the safety monitoring. For most of those topics, we were able to get those in the order they had suggested, and definitely in the one of those listings. But for efficacy endpoints and the safety monitoring, we created multiple listings within each section. So for efficacy endpoints, we created section 8.1 and 8.2 that included both the primary endpoint and the key secondary endpoints. Similarly, for the safety monitoring, the labs, ECGs, vitals, we included it as multiple sub-listings, section 10.1 and 10.2 and so on. Then now that we know what we have to create and what the draft guidance says, then we came up, what was our goal was to use the existing CSR listings. And at the same time, we wanted to automate this process so that, like, it's not just helpful to this our current submission, but in the future in different therapeutic areas or different submission, we should be seamlessly able to use these macros or the programs which we worked on and use those CSR listings and create those 10 oversight topic site listings. So that was our goal. And in the next slide, I'll briefly talk about the workflow. How did we achieve this overall process of using our CSR listings and to create this site level listings by site summary for each of the oversight topics that I just mentioned about. So we took our existing CSR listings, which are in the RTF file. We read those RTF files and we stored in a SAS data set. Once we have those in the SAS data set, we extracted those listings by site and we filtered it out by the site. So for listing 1 to list, for the 10 topics, listing 1 to listing 10 topics, we extracted based on the site 1, site 2, and site 3 until site N. And then we created separate data sets for those. So we iterated the whole process for each of those listings. You filter it out by the site, concatenate with the other oversight topics from the same site, and then we create the listings. So we bundle them once we have those listings from each of those individual sites. So for site 1, we extracted listing 1 to 10 or all the topics that are associated with, and we bundled and we created the RTF file for that one. So now we have for site 1, all the oversight topics, similarly for site 2 and so on. Once we have these RTF files, we created the corresponding PDF files, and then we created one combined PDF file that includes for each of those study, for each of those site, the 10 oversight topics. I'll talk briefly about the details of the technical details, what we did and how did we achieve that overall workflow, which I just talked about. So here is an example we created, like where we had the existing CSR listing 1. And using this RTF file, we extracted our titles from that existing CSR listing. We extracted the footnotes into that. We stored them in a separate SAS data set, both those titles and the footnotes. And similarly, we also extracted the column headers. While extracting the column headers, we needed some additional attributes that would help us to replicate this site level listing in the same format what we created for a CSR listing. So to achieve that, we also extracted the column attributes, which are the column widths and the column justification. And we did the same process for the column values. You take the column values in a separate SAS data set along with the column justification information and any other identifier variables that would help us to make sure we create the listing in the same format what we had it in the existing CSR listing. So as I just mentioned, we had our everything in a SAS data set. We stored our titles, footnotes, column values, column headers. Then we started extracting those from the SAS data set, and we write a proc report SAS code to generate a site level listing. So we extract the titles from that, we extract our footnotes, and we write in our program. In addition to that, we also gave an option to have additional subtitles, like, here we are generating, since we are generating by site, we have included the site ID and the site name in the reports what we are generating for the by site listings. And the next step would be like, using the SAS datasets where we store the column headers along with their column bits and justification. We extract that and we populate into our SAS code where the values will be written down. And finally, we use the column values along with the justification. So by following these 4 steps, we were able to exactly replicate the listings filtered out based on the site, and we created the listings for each of those sites, which were the same content which was as in our CSR listings. Now that we were able to generate these listings, we wanted to again convert them, as I just talked about, to convert into the PDF file. So once we have the listings, all the 10 listings for a particular site, we bundled them to create one RTF file. And we iterated this process for all the sites. We did convert them again into the PDF file using a VB script. And we also created the bookmarks. For the creation of the big bookmarks, we extracted from the RTF file and we created a text file with the 3-level bookmarks. And then using our Adobe plugin tool, we were able to plug in our bookmarks into the final PDF file, which has the same content and style and the format as suggested by the draft guidance of the BIMO, which says the study number by site and the 10 oversight topics. And also we did the QC of these listings to make sure the content matches what we had in existing CSR listings. And the independent QC was done using those combined RTF files. Seems like a seamless process, like the whole idea, the flow went through, like, what we envisioned and what we really wanted, we did achieve it. But there were some inherent challenges as well. One of the most common issue we have noted while working was, like, as in a CSR listing, there will be some set of data for each of those listing components. But once you start filtering it by the sites, some of those listings might not have any data. For example, the most common thing we would definitely see is these inclusion-exclusion criteria or the IPDs. You might have some IE violations or some IPDs. But once you start filtering by the site, most of the sites wouldn't have any of those data at all. So we created an additional proc reports as code so that we populate with a custom message for each of those sites. If there was no data, we were not able to pull up any data for that specific site ID, then we would populate with a custom message saying that no data met the criteria for this listing. The other challenge we had was to make sure, like, how do we use this spanning column headers? So we have to identify this pipe delimited. Once we extracted the RTF, we had this pipe delimited, the way to identify this spanning column headers, and we populated in our proc reports as code. So that concludes the overall process, what we did to create our existing CSR listings into the site level listings as requested by the draft guidance and the technical conformance guidance of the BIMO. While building this overall whole BIMO package, we thought, like, we need to make sure not just within the programming department or a biostats, but we need to have a very strong collaboration working with the regulatory folks and regulatory publishing and the clinical operations, because there are certain aspects of the data we definitely need to work and get, like the site investigator details. We need to work with our clinical operations team to get those level of details. And also we need to, while working on the clean site data set, there were additional, as Olga just talked about, some additional programming considerations that needs to be taken care. Similarly, we are so much used to creating the define XML. This was something we need to revert back and go back to create our define.pdf. So we had some honing hell and glitches, but we were able to work it out and we were able to generate our define.pdf. And one of the neat stories of this whole process was this whole macro which we worked on to generate this CSR, using the CSR listings and generate this site level summary listings. And without an industry defined template, like the way we were used to looking at the views for our CSDRG or ADRG, BIMO RG, we didn't have any template for this. So we came up with our own content where we thought that would be very useful for the reviewers. And we strongly encourage to include the BIMO reviewers guide, given that, like, the BIMO draft guidance states, like, for the documents that are already provided in your BIMO submission package, it is not really required to submit again in your BIMO submission package. So we provided hyperlinks for all those individual sections in the BIMO reviewers guide so that the inspector has one single document with access to all the different type of information through this BIMO Reviewer's Guide. We would like to thank Andy Lish, who was one of our key programmer working on this macro, and the Vertex Programming Management for providing an opportunity to work on this project, and Fuse for providing this platform to share our story. With this, I'll open up for questions.
Thank you, Olga and Raghavender. As you mentioned, we don't have much information out there about BIMO, so that's something that I learned today. Thank you so much for that great presentation. We are now open for question Q&A session for this presentation. We have a question here. Any challenges with populating TRT-EFFR and TRT-EFFS for primary endpoint by site, where SAS errors show up because of low counts at a site?
Fortunately, we didn't have those issues in our data set. We didn't have any low counts, so we were able to work it out. But definitely, if we had our bias stats counterpoint, if we had to run into any issues, but we did not see any in our project.
Thank you. The next question, what is major purpose and challenges of the BIMO listings? How will this help RA for review?
For the inspector, definitely he would like to, if they want to go for a particular site, given that our existing CSR listings are very huge in content and you cannot filter out the data for that specific site. But having these site-specific listings or a PDF document, the inspector can quickly review against the source documents of the site with the CSR, with these site listings which we provide in our submission package.
Thank you. Those are some of our questions. We don't see any more. Is there a group to build more information on BIMO, Raghavender? Like how we have the Fuse working group. Do we have any working groups to build documentation around BIMO?
I would think this is a good opportunity for Fuse to start thinking about, definitely, because that's one place where I always look up for, like, for any industry templates or anything. And we couldn't find any reviewers guide or anything. So we had to really work on ourselves on our end. But we thought sharing this story and definitely something for Fuse to look up to provide a reviewers guide template. Given that this draft guidance was published relatively a couple of years back, I'm sure it's on the cards for the Fuse.
We have a few more questions that came through. Why not use SDTM or ADAM to create the subject level listings?
Yes, definitely that was one of our intent, but sometimes we want to make sure to ensure a consistency between our CSR listings and the site level listings. We thought this is much more streamlined and we need not reprogram or recreate those shells from the stats or anything. We have everything what we needed in our RTF outputs. Why not use them so that we have a consistency between the two sets of outputs?
Okay. The next question, this would be the last one. How do you recommend to QC these listings and data set?
Internally, yes, we did an independent QC where we use these bundled RTF listings for each particular site as a production, and the QC programmer recreated using those SDTM ADAM data sets and made sure he matched the content. So that way we are in a three-way, we are consistent with our data sets with the CSR listings and the bundled site listings.
And I would like to add about clean site dataset. We also have two programmers, independent programmers. One did development and another one QC. So yeah.
Thank you, Raghavendra and Olga. Appreciate great presentation again.